to present on behalf of the Epi4K consortium and collaborators. What I'll be talking about isn't too dissimilar, actually. It's probably more similar than not to what the previous talks discussed, only difference being that we're focusing here on the common epilepsies, but still looking at the contribution of ultra-rare genetic variation to these common epilepsies. So I actually got an opportunity to take some introductory slides out because they'd already been discussed, things like describing exact and ultra-rare variants, etc. I do want to start by acknowledging all the institutes and researchers that are involved. So even though you're listening to my voice, this is the result of work from many individuals around the world. Many of you would have been familiar with some of the earlier work that was done by Epi4K looking at epileptic encephalopathy trios. There are a couple of papers that were published, one in 2013, one in 2014, that looked at trying to identify excess de novo mutations in this patient population. And what we found, particularly within the 2014 paper, where we had about 360 trios that were, and that was a collaboration with the Europeanomics group, published in AJHG for anybody that's interested, was that there were indeed genes that were securely implicated on the basis of excess de novo mutations, and that's nicely illustrated by this plot that Andrew Allen developed on behalf of Epi4K. And what we have here along the red line is your kind of genome-wide correction threshold. And so genes that have more de novo mutations, genes that are above this red line are genes where we see excess de novo mutations when you take into consideration the underlying mutation rate and also the number of trios that were studied. And so through this process, Epi4K, the papers were able to implicate two new genes, being GABA-B and DNM1, and a third gene, ALG13, on the basis of the same residue being repeatedly mutated. Some of the other interesting things that came out of this was, these gene set enrichments, which were discussed within the autism and schizophrenia collections as well. So there was clear enrichment for de novo mutations happening in synaptic transmission-associated genes, genes known to cause seizures when aberrated in mouse models, and also FMRP-associated genes. And based on the 2014 paper at that time, we estimated that about 12% of the trios had an identifiable de novo mutation in one of at the time 26 high-confidence CE genes. And now, since then, actually in the last 2 years, there have been a lot of other genes that have been implicated in epileptic encephalopathy, so that rate has increased as expected and is somewhere upwards of 15% of that cohort now on the basis of new literature. So I mentioned a lot of the epileptic encephalopathies because that will come into play later on in the talk when I talk about some of the common epilepsy results. But the focus today really is on the common epilepsies and some of the work that the Epi4K consortium has been doing in that space. So we've sequenced two collections of broad common epilepsies. First one being a cohort of about 525 unrelated index cases with non-acquired focal epilepsy and a family history of epilepsy. And we've also sequenced about 640 genetic generalized epilepsies, again unrelated index cases with an epilepsy family history. Now, both these cohorts were compared to a collection of about close to 4,000 controls. These were individual sequenced using the same platform, same bioinformatics processes, and they were not ascertained for epilepsy, neuropsychiatric, or any other neurodevelopmental or severe congenital disorders. This cohort of roughly 4,000 samples is quite important because it allows us to estimate what the background variation rate is for a given region, usually a gene of interest. And by being able to sample from this kind of general population background variation rate, it allows us to identify genes where we see excess variation in the case collections. And this is similar to, pretty close to what was described in the previous talks as well in terms of analysis approach. So in terms of qualifying variants, this has been discussed quite a few times. There are many ways to define what a qualifying variant is. But in essence, what you do is you define some kind of category of information that you are focusing on, and then you want to ask whether there is more variants of that fitting that kind of qualifying variant category in your case cohort over the general population background variation controls. And so the two that I'll be discussing here are ultra-rare deleterious, which we've heard about. And this is defined within this data set as external monoallele frequency of 0% based on EVS and exact. So these variants are not present in the EVS or exact data sets. They have an internal test cohort monoallele frequency of about 0.05%. So within the combined case controls, there are allowed to be at least a couple repeat observations. And they're focusing on loss of function and variants predicted to be damaging by polyphen. And then we also did a loss of function specific model where we were a bit more permissive on the monoallele frequency, but focusing really just on those variants annotated as having a predicted loss of function effect. So in terms of the results, when we looked at the non-acquired focal epilepsies, again, this is just 525 cases, but we were able to actually identify two genes that were genome-wide significant. DEPDEC5 and LGO1 were significant after correcting for the roughly 18,000 genes tested. So the adjusted alpha is about 2 to the -6, and both were below that. Both these genes are actually well-established focal epilepsy genes. So in terms of novelty, we're not implicating new genes here. But what's reassuring is that it is clear from these results that this framework, this analysis approach, is capturing true epilepsy risk signal. And in fact, if you look at the top 10 genes in the non-acquired focal epilepsies, the top 5 genes are well-established dominant epilepsy genes. And to put that into context, there are about 43 well-established dominant epilepsy genes, and the likelihood of identifying or having 5 of those 43 take up the top 5 positions out of 18,000 positions is quite unlikely, with a p-value around 5 to the -14. So this really is reassuring, and it does motivate us. And there are studies emerging, such as EPI25 and others, that are interested in sequencing more of these non-acquired focal epilepsies to try and identify which other genes within that are creeping up near the top might break through the genome-wide correction threshold as a result of larger case collections. The other interesting thing within the non-acquired focal epilepsies is that when you focus just on loss of function alleles, what you find is that for gene like DEPDEC5, it is crystal clear that the major signal driving the DEPDEC5 enrichment that we saw in the ultra-deleterious results was on the basis of loss of function alleles. In actual fact, so beyond going a number of orders of magnitude more significant, when you focus back on the missense variants, even those that are predicted to be probably damaging by things like polyphen, there actually does not seem to be any elevated rate of such missense alleles in DEPDEC5 among cases, given what you would expect to see based on the sampling of the general control background variation. And so this has some important implications in terms of interpreting DEPDEC5 variants within the clinics as well. So one of the most exciting things that came out of this actually is that having these data gave us one of the first opportunities to actually really digest what is the relative contribution of increasing monoallele frequency to epilepsy risk within these well-established dominant epilepsy genes. And so we've had similar analyses presented in the previous talks, but essentially what we did is we ran a conditional analysis looking at or asking the question of, after taking into consideration the ultra-rare variation class within these established dominant epilepsy genes in non-acquired focal epilepsy, if we become a bit more permissive and allow even a handful of variant observations among exact, which is 0.005% is understandably still considered quite rare. Do we see an increased enrichment in the case cohort? And in actual fact, based on the current data, there is no evidence to suggest that going beyond ultra-rare variants in the non-acquired focal epilepsies within the known epilepsy genes, that additional epilepsy risk signal is caught by increasing monoallele frequency. And so just to emphasize, this is within a conditional analysis framework, so all together in a multivariate model, and the neutral here is just based on the rate of synonymous variants in the cases versus controls in this set of dominant epilepsy genes. So acting as kind of a negative control there. in terms of the GGE results, so this was a comparison of 640 unrelated index cases with genetic generalized epilepsies compared to, again, the same collection of about just under 4,000 population controls that we had available. Unlike the non-acquired focal epilepsies, there really didn't appear to be any individual genes that broke through the genome-wide correction threshold. There are a few that are creeping up there, and one of the interesting things, though, again, like the non-acquired focal epilepsies, is that, again, we're seeing well-established epilepsy genes making their way to the top. So it's clear that this framework is, for both non-acquired focal epilepsies and genetic generalized epilepsies, the signal-to-noise ratio is fairly good within this framework, and the real signals are emerging among them. And if you take a look at the top dozen or so genes within the genetic generalized epilepsies, you have KCNQ2, GABAG2, and SCN1A, all well-established epilepsy genes. And then you have some others in there, so things like ATP1A3, which is the alternating hemiplegia in childhood neurological disorder gene, and some others in there as well. And you're seeing this trend, and I don't have a plot for it here, but a trend of similar to what was described before, of these intolerant genes based on the ARVIS scoring system, seeming to capture more of this risk signal better within the genetic generalized epilepsies. In terms of gene set analyses, which were touched on in the previous talks as well, the interesting thing about the genetic generalized epilepsies, as I mentioned before on the second slide, is that there is clear enrichment of known epileptic encephalopathy gene overlap for the GGE population. And in actual fact, even if you look at the other gene sets that we had implicated within the epileptic encephalopathies based on de novo mutation excess, within this study, case control study, there was also an excess of FMRP-associated synaptic transmission and mouse seizure aberrated genes within the genetic generalized epilepsy cohort. So this is quite interesting because it does begin to suggest that a lot of these genes that are relevant to epileptic encephalopathies could also be very relevant to these more mild representations of epilepsy that fit into this genetic generalized epilepsy syndrome. And again, this is just a neutral variation enrichment to show that there is no excess, there's no enrichment of synonymous variation within these gene sets, comparing the case to control cohorts. Within the genetic generalized epilepsies, again, doing a conditional analysis, based on the current data, we still, within the genetic generalized epilepsies, don't see any evidence for an attribution of additional epilepsy risk on the basis of increased monoallele frequencies beyond those that are ultra-rare variants based on these dominant epilepsy genes. So in kind of summation, when you look at these two groups, it's quite clear from the genetic generalized epilepsy data that it's unlikely that any individual gene will explain greater than 1% of the broad GGE population, and it's likely from the GGE data that there is going to be much greater locus heterogeneity. And what's very encouraging though is that it's very clear from the data and from the gene set analysis in particular that there is clear evidence for ultra-rare variants contributing to risk for even this common complex epilepsy population. And so, we're really encouraged by this, and things like Epi25 and so forth that are seeking to sequence more and more of the GGE population will hopefully be able to push more of these GGE risk genes that are explaining smaller percentages of the overall population through the genome-wide significance correction threshold. In somewhat contrast to the GGEs, for the non-acquired focal epilepsies, it's interesting to note that if you take just the top 5 genes, they do appear to account for approximately 8% of the familial non-acquired focal epilepsy collection. And after taking into consideration the background variation rate of qualifying variants in these genes, each one of these genes do appear to contribute to greater than 1% of the population individually. So it's, whether this pattern will hold up with the new non-acquired focal epilepsy genes that will turn up is to be seen. But it is an interesting contrast to the GGE results. And just as a kind of a final slide, what really came through this is that for a long time within epilepsy genetics, we'd thought about an individual epilepsy gene and an individual epilepsy syndrome, And there were a few examples where this wasn't the case. So, example being like SCN1A, where we know it causes Dravet syndrome, which is a very nasty, severe epileptic encephalopathy disorder type, and then it can also cause GEPS plus. And however, that was thought to be an exception. And what's coming out of these data is that, at least within the genetic generalized epilepsy data, is that it appears that spectrum genes and more and more of these genes are likely to be contributing risk to both the severe early onset epilepsies and also these more common, more milder presentations such as GGEs, or relatively milder compared to the epileptic encephalopathies. And finally, again, it's been emphasized over and over throughout the talks, but it really is clear within the epilepsies as well that it's these ultra rare variants that are most relevant. And when you take into consideration that it's these ultra rare variants that are most relevant, and also the strong overlap with genes that are known to cause severe epilepsies, it really is exciting, given there is a lot of effort going into the severe epilepsies in terms of the epilepsy precision medicine paradigm of targeting treatments to these genes. And the fact that there are shared genes within these severe and milder epilepsies indicates that a lot of these, potentially these therapies and management that comes out of the epileptic encephalopathies can be translatable to some of the more common epilepsies. And so, again, just finishing up on the acknowledgements, and happy to answer any questions.